Welcome you all to my YouTube channel. Today, we will study about the topic mechanism of apoptosis. Apoptosis occurs when certain enzymes called as caspases are activated. Activation of these enzymes depend on BCL family of proteins. This BCL family is divided into three groups: anti-apoptotic, proapoptotic, and sensors. This division is based on their function. Occurrence of apoptosis depends on the presence or absence of survival signals. When survival signals such as growth factors are present, they induce production of anti-apoptotic proteins such as BCL2, BCLXL, and MCL1. These proteins are present in the outer mitochondrial membrane and they make it impermeable. So, leakage of mitochondrial proteins such as cytochrome C is prevented. When cytochrome C is released into cytoplasm, it initiates the suicide program of apoptosis. Let us see what happens when survival signals are lacking or some sort of cellular stress occurs. When a cell is damaged or stressed, BH3 only proteins or sensors are activated. Members of this group include BAD, BEAM, BID, PUMA, and NOXA. These proteins regulate the balance between anti-apoptotic and proapoptotic proteins. During periods of cellular stress, they activate the proapoptotic proteins. Proapoptotic proteins are back. The function of these proteins is to increase the permeability of outer mitochondrial membrane. Though the exact mechanism through which they permeabilize the outer mitochondrial membrane is not known, according to one model, it occurs through formation of channels. So, now since the permeability of outer mitochondrial membrane is increased, cytochrome C can leak out into cytoplasm. So, what will it do there in the cytoplasm? It binds to a protein called apoptosis activating factor 1, which forms a wheel-like structure. This complex between cytochrome C and apoptosis activating factor 1 is called apoptosome, and it binds to caspase 9. Caspase 9 is the initiator caspase. It begins activating adjacent caspase 9 molecules by cleaving them. Along with cytochrome C, other proteins leak out from mitochondria. These are smack diablo. They bind to cytoplasmic protein called as physiologic inhibitors of apoptosis. As the name suggests, they inhibit apoptosis by blocking the activation of caspases. Thus, they keep the cell alive normally. But when they bind to mitochondrial protein smack diablo, they get neutralized. This leads to propagation of caspase cascade. This pathway, which occurs due to leakage of cytochrome C from mitochondria, is the intrinsic pathway. We have understood about how the initiator caspase 9 gets activated. Now, we have to study about the execution phase of apoptosis. But, before that, let us study about the extrinsic pathway of apoptosis. The plasma membrane of cells contains several receptors. We will study about the death receptors here. The important ones among death receptors are type 1 TNF receptors, TNFR1, and FAS, CD95. These receptors contain a cytoplasmic part called as death domain. It delivers apoptotic signals to cytoplasm. There is ligand for FAS, called as FAS ligand. It is present on cytotoxic T cells and T cells which recognize self-antigens. Thus, it helps in elimination of self-reactive lymphocytes and virus-infected cells. So, when FAS ligand binds to FAS, 3 or more molecules of FAS come close to each other. So, their cytoplasmic death domain is also close together. This forms a binding site for an adapter protein. This also contains a death domain, and the name for this death domain is FAS-associated death domain. This FAS-associated death domain binds in active form of caspase 8 and caspase 10. Many procaspase 8 molecules come close together, and they start cleaving one another. So, now there are numerous active caspase 8 molecules. This leads to activation of executional caspases. Now, we will study about execution phase of apoptosis. So, activation of caspase 9 occurs through intrinsic pathway, and activation of caspase 8 and 10 occurs through extrinsic pathway. These initiator caspases now activate executional caspase 3 and caspase 6. These cleave inhibitor of cytoplasmic DNases. So, the DNase become active and start cleaving DNA. These executional caspase start degrading nuclear matrix and cause fragmentation of nuclei. Also, there is formation of apoptotic bodies, though the exact mechanism through which these apoptotic bodies and cytoplasmic plebs are formed is not known. These apoptotic bodies attract phagocytes, which eat them up, before inflammation sets in. This is how the body gets rid of damaged cells. If you like this video, please do like, share, and subscribe.